Good morning, everyone. I'm Zengyi Shao from Iowa State University. I'm associate professor from Chemical and Biological Engineering Department. My group joined CABBI about 1.5 years ago. I appreciate the opportunity to represent the conversion theme today to deliver a Bioenergy 101 talk today. Leslie guided me that a 101 presentation is aimed to give a CABBI researcher a quick view about the topic area outside their main focus that help inform and could potentially lead to future collaborations. And therefore, today's talk will not focus on too much technical detail. Instead, I will explain the rationales for us to decide on which products and which hosts we should target on in the next few years. As many of you already know, conversion theme chose to focus on a few yeast species as the host. Some of you may wonder what made us choose yeast initially instead of bacteria. In the context of a DOE interest, yeast do not have a phage infection issue. Some of them are resistant to acidic and alcohol condition. As you already know, some of our target products are organic acid and alcohols. And very importantly, yeast biomass can be separately sold as animal feeds, further increasing the profit of the whole production process. On the other hand, they are much more sophisticated than prokaryotic hosts. Regulatory controls exerted at different levels often lead to low expression, gene expression levels, and the production titer at the beginning development stage. And beyond the compounds interested by DOE, some of us in the conversion theme chose to work on yeast in parallel projects because yeast are better suited hosts for producing some high-value compounds currently extracted from plants due to their ability for P450 enzyme expression. And lastly, beyond working as a production host, yeast are simple but fast-growing testbed for studying chromatin biology in higher eukaryotes and models for some human disease. We chose these three categories of compounds as a target. For the sake of time, I will focus on just a few examples for today's talk. The first group, lipid-based chemicals. They include fatty alcohol and wax esters. They are made from this simple metabolic intermediate acyl-CoA, shown here, through these 1-to-2 enzymatic reactions. The enzyme FAR convert acyl-CoA to fatty alcohol, which is mainly applied as a detergent and a surfactants, as well as a skincare product additive because of their emollient and occlusive characteristics, altogether with around $1 billion market currently. Oh, I forgot to show this. Okay, fatty alcohol can then react with another acyl-CoA molecules to form long-chain wax ester, catalyzed by this wax ester synthesis. Wax esters can be utilized as a premium lubricants and a skincare product additive due to their similar structure with human skin. Our conversion theme chose to engineer two oleaginous yeasts, Yaruvia lipolytica and Rhodosporidium pteryloides, because of their superior native ability to accumulate TAG at high content. Because TAG are also derived from these acyl-CoA molecules, so we reason that these two yeasts should provide abundant precursor for fatty alcohol and wax ester biosynthesis. The second category of compounds is organic acids, and the citromalate is one of the representatives we chose. Citromalate can be converted from pyruvate and acyl-CoA, shown here, catalyzed by the enzyme encoded by SIM-A. After bioproduction, citromalate can be further converted to MAA through simple chemical conversions, and finally polymerized to be PMAA, which is a lightweight, transparent, shatter-resistant material currently used for constructing residential and commercial aquarium and making the lens of exterior light of automobiles and hourglasses. Another organic acid the conversion theme is trying to make is a 3HP, which is a precursor of a fully biodegradable polymer. For both the citromalate and the 3HP, we chose Escherichia orientalis as the production host due to its high acid tolerance. As you can see here, even at 80 grams per liter, the strain can grow very well, right? Yeasts group have screened 170 essential or enterlist strain and successfully identified the ones demonstrating hypertolerance towards high concentration of lignocellulosic inhibitors, but leaving little drug resistant to the environment. However, despite their unique and superior metabolic and physiological capabilities, compared to model yeast Saccharomyces cerevisiae, we are still short of engineering tools, and our understanding for their genetic and biochemistry is little. So in general, stable episomal expression plasmid and precise genome editing tools are the two foundational technologies for strain engineering. In Saccharomyces cerevisiae, the model yeast, we have ARS centromere-based low-copy plasmid and a 2-micron-based high-copy plasmid, and very efficient genome editing technology based on homologous recombination. However, for majority of the non-conventional yeasts, there is usually no stable episomal plasmid available. What is even worse is that many of them use non-homologous adjoining instead of homologous recombination for chromosome double-strand break repair, which will make precise genome editing efficiency extremely low in this species. So as you can see here, without using CRISPR editing, the precise genome editing efficiency is less than 1% for RT and 5 to 10% in Yaruvia. Just a second. The alternative strategy nowadays is to use CRISPR genome editing, and the conversion team has made such a tool available for Yaruvia and Escherichia with very high efficiency. For rhodosporidium, due to the dominancy of NHEJ mechanism, so far, the HR-based integration efficiency has only been improved to 20%, but this comes from less than 1% efficiency as the initial point, which we deem as a milestone. The applications of such a tool include to perform targeted deletion, marker recycling, and targeted integration. Here, I want to point out that for targeted integration, actually, we don't have the knowledge about which loci are good integration and expression hotspot. And very often, a good integration hotspot is not equal to an excellent production hotspot. This is because a highly efficient integration spot may come from easy accessibility of genome, but its local genomic environment might not yield ideal expression dynamic and therefore high production of the desired compounds. To obtain such a fine-tuning pathway expression tools, we then leverage both non-homologous adjoining and transposon-mediated integration mechanisms to hop the pathway randomly into the genome, and then screen the beneficial loci whose spatial temporal genomic architecture yield favorable expression dynamic for the desired pathway, leading to high production of the target compounds. As a team, the conversion team also focuses on automation of the genetic manipulation tools I just mentioned, and then develop of a high throughput analytical tools to facilitate screening, as well as development of system biology and artificial intelligence tools to facilitate biochemical engineering understanding. We also have a catalysis subgroup to further diversify the product portfolio, and the deconstruction group, a subgroup to provide us the raw hydrolyzate as the feedstock, better making our process better mimicking the industry production. So for that, I will stop here, and please feel free to ask me any questions.